Good morning. So, we have finished the endocrinology. So, today we are starting the physiology of reproduction. So, for the first, we have to know about the sexual growth and development. So, the in human embryo, sexual growth involves two processes. One is sex determination and the other one is sex differentiation. We all know that humans have 46 chromosomes, out of which 22 pairs of autosomes and then 2 sex chromosomes. So females will be having 46 chromosomes, 46 chromosomes, and they have the sex chromosomes XX, and males will be having XY. So first we'll see how is the sex of an individual is determined, or is the sex determined by the presence or absence of the Y chromosome. So I already mentioned that the in human embryo sexual growth involves two processes: sex determination and sex differentiation. So the answer for the previous question, that is how the sex is determined, was provided by the individuals, okay, resulting from the non disjunction of the sex chromosomes, so that the abnormalities, okay, during the process, what will happen, some they will be having 45 chromosomes and some they are having they have 47 chromosomes. By this abnormal chromosomes, so it shows that the presence of the Y chromosome is necessary for the male sexual characteristics to develop. So first we'll see, so this is the normal female, that is having 46 XX. Okay, so first we'll see the sex determination. It is also known as genetic differentiation. It refers to the genotype of the fetus, whether it's a male or a female. So, human chromosomes, each cell except ovum and sperm in a normal adult male and female possesses 46 chromosomes, 44 autosomes plus 2 sex chromosomes. The females possess 44 plus XX, and then the males possess 44 plus XY. The mature male gametes are called sperms and mature female gametes are called ova. Okay, so here each ova, it contains 23 chromosomes. Since the primitive female germ cell oogonia, from which mature ova are formed, contain 44 plus XX chromosomes, so each ovum will be getting 22 plus 1 sex chromosome. Okay, 22 plus X. The primitive male germ cells spermatogonia, from which mature sperms are formed. Okay, the primitive male germ cell that contains 44 plus XY. So when it undergoes from this spermatogonia, so this divides it into two. So one will be having 22 plus X and the other one will be having 22Y. So this is about the human gametes. So genetic sex determination of the embryo occurs during fertilization. That is the, the penetration of the ovum by a sperm. So when an ovum, okay, which is having 22 plus X is fertilized by a sperm containing 22 plus X, the resultant will be 44 plus XX. So that is the female genotype. When an ovum 22 plus X is fertilized by a sperm containing 22 plus Y chromosome, then the resultant zygote will be 44 plus XY. That is a male genotype. So this is about the sex determination. This is how a sex will be determined. Next we'll see how it is differentiated. Sex differentiation. So this is an important diagram. So before explaining the diagram, I just wanted to mention few things. That is, after fertilization, that is when once the sperm enters into the ovum, the normal sex differentiation in the embryo proceeds sequentially. So the stages of sex differentiation are gonadal differentiation, genital differentiation, and psychological differentiation. Gonadal differentiation, in the sense, gonadogenesis refers to the formation of the testes in males and ovaries in females, whereas genital differentiation means this is the phenotypic sex differentiation, that is refers to the differentiation of internal and external genitalia, whereas psychological differentiation, in the sense, normal sexual behavior in an adult male and female, and this is determined by the effect of androgens on the development of brain in the embryonic stage. So these are the stages of sex differentiation. So first we'll see this diagram. So this is the diagram which shows the Wolffian duct and then the genital ridge, and then in this genital ridge, okay, this is the important one, or the urogenital ridge is the site where the gonads develop. You can see the diagram, the genital ridge. So this is the site where gonads develop. Here what will happen, the primordial germ cells, they migrate into the genital ridge and forms the bipotential gonads. So the bipotential gonads, these are also called primordial or indifferent or ambisexual gonads. So up to 6 weeks of gestation, the bipotential gonads are identical in both the sexes. That means up to 6 weeks we can't identify whether it's a male or female. So this bipotential gonads, it consists of a cortex, it consists of a medulla, and then primordial germ cells. So if it's a male, how it will differentiate into testes. If it is a female, how it will differentiate into ovaries, that we will see. So testicular differentiation from a bipotential gonad. In genetic males, embryo, the bipotential begin to differentiate into the testes at approximately 6th week. So at about 6th week of gestation, the testicular differentiation begins with the appearance of the primitive seminiferous cords from the germinal epithelium covering the medulla of bipotential gland. See, the bipotential gland is originated from the genital ridge, and this bipotential gland is having a cortex, medulla, and then the cortex, medulla, and the primordial germ cells. So at about 6th week, if it is a male, so what will happen? The testicular differentiation starts with the appearance of primitive seminiferous cords from the germinal epithelium that covering the medulla of bipotential gland. At about 8th week of gestation, these Leydig cells appear in the interstitial spaces of seminiferous tubules and continue to proliferate. The Leydig cells are derived from the sex cords that are not canalized. The membrane of the Leydig cells, these has receptors for human chorionic gonadotrophin. At about 9th week of gestation, the Leydig cells synthesize and secrete testosterone in response to the HCG by the placenta. So at about 5th week of gestation, there occurs descent of testes through inguinal canal into the scrotum. So this marks the final stage of testicular differentiation. In this, role of Y chromosome in testicular differentiation. You should know that what is the role of Y chromosome in testicular differentiation. On Y chromosome, we have two genes, one for the testicular differentiation and another for the formation of a substance called Mullerian duct inhibitory substance. So the gene which is responsible for the testicular differentiation is called SRY gene, that is the sex determining gene of the Y chromosome, that in short form it's called SRY gene. So the SRY gene encodes the testis determining factor. So this gene, from this gene, the testis determining factor will be formed. So this produces the Sertoli cell differentiation. And one more thing, it is having a gene which forms Mullerian inhibiting substance. So if this substance is produced, it inhibits the Mullerian duct. So when there is inhibition of the Mullerian duct, then there will be the result will be male. Next we'll see the ovarian differentiation. The in genetic female, okay, how the bipotential gonads, if it is a female, how it is differentiated. So here by about 10th week of gestation, so this is what I mentioned the previously. Okay, this is the diagram of the testes, development of the testes. So now we'll see the ovarian differentiation. In genetic females, it is 44 plus XX embryo. By about 10th week of gestation, ovarian differentiation occurs in the absence of testicular testis determining factor. That means when there is Y chromosome, okay, so Y chromosome is having a gene which produces the SRY, that is the gene responsible for the testicular differentiation, and it's having a gene for the Mullerian inhibiting substance. So if there is no Y chromosome, okay, if it is a female double X XX, so what will happen? Then there will be absence of these two factors. So in the absence of these two factors, then what will happen? Oh, there will be differentiation, ovarian differentiation. So the ovaries develop on each side from the cortical region of the bipotential gland. The coelomic epithelial cells proliferate and form granulosa cells which surround the germ cells and commit them to oocyte formation. During 11th to 12th week of gestation, oogonia undergo meiotic division to form oocyte, which is the end point of ovarian differentiation. So this is about the gonadal differentiation. So next we'll see the genital differentiation. So this is the thing we should know, that is the Mullerian duct and the Wolffian duct. So Wolffian duct is responsible for the formation of males, whereas Mullerian duct is responsible for the formation of the genitalia in females. So next we'll see the genital differentiation. The internal genitalia differentiate from the neutral sex anogen, which develops during the 6th week of gestation along with the development of the bipotential gland. So this is the diagram. You can see the Wolffian duct as well as the Mullerian duct, and then you can see the gonads as well. So first we'll see differentiation of male internal genitalia. So in the genetic male fetus, so the testosterone which is secreted by the Leydig cells that stimulates the Wolffian ducts to form the epididymis, vas deferens and seminal vesicles. And we mentioned that Mullerian inhibiting substance which is produced by the Y chromosome that causes regression of the Mullerian duct, okay, that is by apoptosis. So there will be regression of the Mullerian duct. So this is about differentiation of male internal genitalia, whereas differentiation of female internal genitalia, so this is when there is absence of the Y chromosome, there will be absence of MIS, that is Mullerian inhibiting substance. So what will happen? The female duct, Mullerian duct, proliferate and that form the oviduct, uterine tubes, uterus and upper 2/3 of the vagina. So this is the diagram showing the differentiation of the genitalia. So the first diagram, the males, so there will be regression of the Mullerian duct, degeneration of the Mullerian duct. So the Leydig cells stimulate the Wolffian ducts to form the epididymis, and then the vast difference and seminal vesicles, whereas the in the female internal genitalia, so when there is absence of Mullerian inhibiting substance, okay, so what will happen, the Wolffian duct will not be there. So the Mullerian duct, it forms, proliferates and forms the uterine tubes, uterus, and then ovaries. So this is about the differentiation of the internal genitalia. So this is the one, that is SRY. So SRY gene is responsible for the formation of the male gametes and for the males. Okay, that means when there is SRY gene is present, so this gene is responsible for the production of the testes. So when the SRY gene is absent, that means when the Y chromosome is absent, then what will happen, that produces the ovaries. So this is about the SRY gene. It's found on the Y chromosome. In the cell, it binds to the other DNA and distorts its shape. This alters the properties of the DNA and likely alters the expression of a number of genes that leads to the testis formation. So if a SRI gene is present, that forms the testis. If a SRI gene is absent, that means if Y chromosome is absent, then there will be formation of ovaries. This you should know. Then so this one point regarding the during the development, if there is a neonatal male, okay, he will develops into adult male with male behavior. But if there is any castration at the critical during the development, but if castration at critical period, what will happen during the development, what will happen if there is removal of the testes, so he may be a feminized male with female behavior, and in neonate female, she develops into adult female with female behavior. But if you administer some amount of testosterone, okay, then what will happen, that females will be masculine and she will be having the male behavior. That's an applied aspect. Okay, and then we should know one point regarding the differentiation of external genitalia. So the external genitalia in both sexes, it develops from the common anal gen, okay, so which is the urogenital sinus, which are the urogenital sinus, the genital sinus, the genital tubercle, and then the genital swelling. So external genitalia develop from the common anal gen, which are the urogenital sinus, the genital sinus, genital tubercle. In male fetus having the functional testis secretes the testosterone. So the external genitalia acquire male characteristics by the 5th month of gestation. In female fetus, because in the absence of any hormone, external genitalia differentiation occurs along the female line. So this is about the genital differentiation. So the applied aspects will be the next. So applied aspect I will tell in the next class.